Hi friends, so in this video we are going to talk about ketogenesis. So as the name suggests, genesis means formation, which means the formation of ketone bodies. So please make sure to like, share and subscribe my channel. So what are ketone bodies and why they are important and how they are generated? So ketone bodies is the water soluble compounds that are produced when fats are broken down. So these ketone bodies are important during the condition like starvation and when fats are broken down, like during starvation you need energy. So one of the source that comes into play is the fatty acids that are present later stored in the adipose tissue, and then they mobilized these fats to be broken down and forms ketone bodies. And these ketone bodies, there are specific organs which can produce these ketone bodies, and then these ketone bodies can travel to different other tissues where these are not produced and can act as a source of energy. And so there are basically three types of ketone bodies: acetone, acetoacetate and beta-hydroxybutyrate. And in my later video I will talk about how they are produced. So here you can see the structure of acetone and acetoacetate and beta-hydroxybutyrate. So acetoacetate is the major ketone body that produce acetone and beta-hydroxybutyrate. Uh, so as I told you, like they are produced in specific tissue, so they are majorly produced in the liver because the enzyme that is the enzyme that is required to produce these ketone bodies is present only in the liver, which is HMG CoA lyase. So we will talk about later how this enzyme works. And the ketone bodies, they are mainly produced in the liver, and from the liver they get transported to other tissues where oxaloacetate or beta hydroxybutyrate, which is one of the ketone body, they can be converted to acetyl coenzyme A. And if you know, and from my previous video, that this acetyl coenzyme A get feeds up into the TCA cycle. So how this acetyl, like what is the source of this ketone body is acetyl coenzyme A. So in my previous video, I have talked about beta oxidation, which is the breakdown of the fatty acids and releases the acetyl coenzyme A. So this is the continuation of that cycle where the acetyl coenzyme A is produced by the beta oxidation in the mitochondria, and then two acetyl coenzyme A, they condensate, like they come together with the help of this enzyme acetoacetyl coenzyme A synthase. They form acetoacetyl coenzyme A, which gets form HMG CoA. So this acetoacetyl coenzyme A is a 4 carbon compound, and then HMG CoA is a 6 carbon compound, because in this acetoacetyl coenzyme A, another acetyl coenzyme A from this, they get condensate and form 6 carbon compound, which then gets broken down, and one acetoacetyl coenzyme A is released and acetoacetate is also released. So if you remember from my previous slide, acetoacetate is one of the major ketone body, and then this acetoacetate, it can be converted either to acetone or beta hydroxybutyrate by the action of different enzymes. So this is how this reaction process. So you have one acetyl coenzyme A, and then there is another molecule of acetyl coenzyme A. With the help of thiolase, this thiol group, like here, is broken down, and this acetyl group get condensate with this acetyl coenzyme A and form acetoacetyl coenzyme A. And now this acetoacetyl coenzyme A, another acetyl coenzyme A group binds and releases this CoA SH group and forms the 6 carbon compound. So here you can see, with the help of this enzyme HMG CoA, which is beta hydroxy beta methyl glutarate, so it's a big name. So now this CH3CO, it get So this compound comes like this molecule of acetyl coenzyme A condensate with this molecule of the acetyl and form this beta hydroxyl group, right? And then we see this compound form, which is called HMG CoA. And then this HMG CoA, with the help of this enzyme HMG CoA lyase, which I have talked is only present in the liver. That's why these reactions happen in the liver. It gets broken down to acetyl coenzyme A, which get released and can be used again in this reaction, and to acetoacetate, which is a 4 carbon compound. And then this acetoacetate, by the decarboxylation, like releases a CO2, it can be converted to acetone, or by the dehydrogenase, it can be converted to beta-hydroxybutyrate, in which an H2 group is added here. So acetoacetate is the major ketone body which can be converted to acetone and beta-hydroxybutyrate. And then this beta-hydroxybutyrate can further move to different tissues and release acetyl coenzyme A, which gets feed into the TCA cycle. So what we have talked about is in the liver, in the adipose tissue, the fatty acids are broken down, and these fatty acids get released in the blood, right? And these fatty acids can be taken by liver or by other tissues as well, and these fatty acids get broken down to acetyl coenzyme A with the help of beta oxidation of fatty acids that I have talked in my last video. And then this acetyl coenzyme A, it can either feeds the TCA cycle and releases CO2 and gives 36 molecules of ATP, or this acetyl coenzyme A, in case of starvation condition, and when the enzymes are especially in case of starvation condition, when the body needs energy, it gets converted to ketone bodies. And then these ketone bodies moves to the blood, and then they can go to different tissues, like they can go to the kidney. So when people have like excess of ketone bodies, this is what is called as ketonuria, which means that ketone bodies are released in the urine by the kidney, or they can be released as the smell of acetone, like they are exhaled from the lungs. So you can feel it when someone has excess of ketone bodies. You will see it either in the urine or from their smell. You can like a person can observe it, or it can go to other tissues like muscles where ketone bodies are not produced, and then ketone bodies can be broken down and release acetyl coenzyme A, which gets further into the TCA cycle and releases energy. So this process is very important for the energy sources during starvation condition. And then, what factors regulate ketogenesis? So, as I have talked about, the source of ketone bodies is the fatty acids. So, it depends on the fatty acid mobilization from adipose tissue. Um, so, insulin, which is released when you are eating or when you have enough energy available, so it inhibits lipolysis, because lipolysis get activated only when you are in starvation condition or you need energy. But when you are full and you are eating, then insulin inhibits lipolysis, while glucagon, which is a source which is an hormone that activated during starvation and to maintain the blood glucose level. So during that case, lipolysis get activated. So it depends on the ratio of insulin to glucagon, the amount of fatty acid activation, mobilization activation. And then excess of acetyl coenzyme A, if you have excess, it gets channeled into the ketogenic pathway, because then you have excess and then you reallocate it to different pathways, and one of the pathway is ketogenic pathway. So I hope you like it, and please like, share and subscribe my channel. If you have any doubts, please let me know in the comment section. Thank you.